Thank you, Kate.
Thank you very much, Dani, for agreeing to give a Build a Cell seminar, and you can get started.
Thank you, Kate. Thank you for the invitation. It's a pleasure to be here with the whole Build a Cell community. I'm Daniela Bittencourt. I'm from Embrapa, Genetic Resource and Biotechnology, which is a research company mainly focused on agriculture. It is vinculated to the Minister of Agriculture here in Brazil. And I'm also research associate at JCVI, where I've been as a visiting scientist for a couple of years. And I still continue with the collaboration with John Glass specifically. So I'll talk about what we have been doing these last years regarding the minimal cell mostly. So we are using it as a tool for specific purposes. But first, I would like to actually to start showing this video. This was developed by Tim Davidson, which is a programmer from Canada. So here he's showing is actually it's a how can I say a model from Mycoplasma genitalium. Mycoplasma genitalium is it's the family, it's a species from the Mycoplasmas family, which is the minimal cell was based off. The minimal cell was designed based on the genome of Mycoplasma mycoides. And actually this model is very, very similar to the minimal cell because it's round, it has a round shape, different from actually for the original Mycoplasma genitalium, which doesn't have specifically this shape. So that's why it reminds us a lot of the minimal cell. But what I want to show you actually, it's what I want to call more the attention for you, it's actually for the membrane of the cell. So this is, let me see if I can start the video again. So it's the membrane. So if you see here, we can actually realize that there are a lot of different proteins and transporters like these ones, which are very, very important for the whole control of the cell. So the whole control of the cell, mostly for osmotic control and also for all the how can I say the interactions with the cell with the external environment of the cell. So and of course the membrane is also a very important organelle. Without the membrane there is no cell. So if you want to understand and how can I say, if you want to fully understand the basic functions that are necessary to build a completely synthetic life, I believe that we have to fully understand the membrane, how it works, how are the key components that we must have to make an appointment on account in order to build a completely synthetic cell. So what we have been doing actually, let me see if I can pass my slides now. I think, how can I move this? How did I do the last time? Okay, now I got it. Sorry, guys. This is not working the way I was hoping for, but that's okay. So talking about the minimal cell, as I said, the minimal cell was developed based on the genome of Mycoplasma mycoides. And it was through that process of genome minimization. And now this cell has almost the half of the genome of the original wild type Mycoplasma mycoides. And it holds specifically the main genes that are responsible for the main functions of the cells, the functions that we say that are essential for it to be considered an organism, a live organism. So we can say that the minimal cell is the hydrogen atom of biology. And because of this simplicity, this is a very, very interesting chassis to develop different kinds of metabolic studies to try to understand at least the principles of life. What is really necessary for us to consider an organism as alive? So for the construction of the minimal cell, it was first built by JCVI, the cell JCVI-syn1, which was very, very similar to the wild, which was very, very similar from the wild type Mycoplasma mycoides. It has almost the whole genome that from this species. And then afterwards, after process of genome minimization, it was built the JCVI-syn3.0. To build these cells, they had a very, very long work. They at the time they were capable to synthesize these small synthetic oligonucleotides, which they were assembled together in larger cassettes, and then through homologous recombinations, they were inserted into yeast. And then after this, it was performed genome transplantations between yeast and a recipient cell, which in this case was Mycoplasma capricolum, another Mycoplasma species very phylogenetic related to Mycoplasma mycoides. And then we were capable to develop the synthetic cell. So, and after the genome minimization, the second round of trying to assemble a smaller genome, JCVI acquired the Syn3.0, as I said before, almost with the half size of the original genome. But interesting, what happened is that when they first built the JCVI-syn3.0, it had actually unexpected morphologies. It didn't show a very regular cellular shape. It had some kind of vesicles, or if you can see here, we have DNA hush staining the DNA, and here the membrane are stained. So you can see different size of cells, and even the content of DNA is kind of weird. So we realized that those cells weren't actually dividing the way we were expecting. Neither their morphology was the way we were expecting. And they were like slower cells to grow. They were very, very sensible. So it was performed a study to identify which genes could be put back in the Syn3.0 to bring it back, this morphology, the regular morphology that we were expecting. And at the time, it was done by introducing 19 non-essential genes back to the JCVI Syn3.0, which brought us to JCVI Syn3A, which has a normal morphology, it has a normal division, so it's an easier, how can I say, it's a happier cell, and it's an easier cell for us to work with. And in order to be capable of bringing back those genes and also work with other possibilities and try to understand other kind of genes, it was developed the JCVI-syn3B. So it was added, it's the same one as JCVI-syn3A, but the difference here is that it has a Krylov site for homologous recombination. So in this sense, this cell, JCVI-syn3B, is actually the one that we use for these studies in order to understand the functionalities of different genes, and also trying to put back other metabolic pathways or even specific genes, so you could change or improve, let's say, the capacities of the minimal cell. But thinking about membranes, okay, and also thinking about the way that the Syn3.0 was morphologically and how it used to divide. So it seems that the minimization process, of course, it also affected the membrane and also its composition. So, and then it came up with a question. So how did the minimization affect JCVI minimal cells' capacity to interact with mammalian or human cells even, and the immune system? Because I'm not sure if I mentioned, but Mycoplasma mycoides actually is a pathogenic cell. It causes disease in goats. So And through evolution, it also the wild type Mycoplasma mycoides developed very, very good systems to avoid the immune responses, and also that there is no cell wall, low GC content, minimal capsule. So a lot of things that we are aware of and we know that are capable to actually induce immunological response are not present in the minimal cell. So there is no capsule, and 80% of the proteins on the surface of Mycoplasma mycoides are not present in the minimal cell. So what happened with this? Does the minimal cell is an inert cell, like doesn't it lost completely its capacity to contaminate a cell or even its pathogenicity? So and then a couple of years later, in 2021, Yanagihara group in Japan actually studying MBA gene, which is considered to be responsible for the attachment of urea plasma cells to mammalian cells, was introduced in the minimal cell syn3B. And here in this study, he just showed that the minimal cell actually wasn't capable to to attach to the HeLa cells. But when they had the MBA gene in there, actually they recovered the capacity of the minimal cell to attach to HeLa cells. So yes, this is actually one of the first questions answered. Okay, the minimal cell lost its capacity to attach. But what about the immunity response? Here we can see the JCVI-syn1. Oh my gosh, now I do not have my videos, do I?
The videos are not playing. Oh, they just started.
Okay, let's see. Okay, so here we have GAJAK60 cells, which are neutrophil-like cells cultivated together with the syn1. You see here that the cells actually, the neutrophils are kind of crazy looking around and trying to reach for the syn1s. In the opposite way here, for the syn3, here is our neutrophil cell. It's kind of okay. You were here. I don't bother about you. In a couple of seconds, it will show up here up again. So they are not as so excited, let's say, in the presence of JCVI-syn3 as they were for JCVI-syn1. So it seems that actually, unlike syn1, syn3A was largely ignored by the human phagocytic cells. So in this sense, we decided to look for which are the genes that are responsible for these characteristics, which genes that are present in the Mycoplasma mycoides and even in the JCVI-syn1 that are capable to make them infectious, let's say, that they're made to survive in co-culture with cells that are not present in the syn1. So what we've done was to look it up for different cells that had the different size of genomes that were built during the minimization genome process. And then we find out two genomes, which was syn11F and the RDG 1.2. And actually what we found interesting and actually was what was actually missing on this genome, and in the case of syn1F, we had only these genes missed, was this cluster from the 179 up to 186 genes. And what is the most interesting in this case is that we found that mostly the first ones, the 179 and 180, they encode for lipoproteins, which are well known to have a central role in interactions between mycoplasmas and eukaryotic cells. And also the other genes, 183, 84, 85, and 86, they are also are related to, they have roles in cell metabolism and also and the metabolism of carbohydrates, which are also essential for host colonization and pathogenesis of bacterial pathogens. So we said, okay, these might be our candidates, and probably they are the responsible for this. And well, we decided then to do different constructions, one only with the clusters with the lipoproteins itself, and another construction with the full set of the 8 genes. And what we found actually in doing microscopy fluorescence, here we can see JCVI-syn1 and each area attached to HeLa cells after 48 hours of culture. But then when we compare it with the other clones, JCVI-syn3B and the other 2, here we have JCVI-syn1. B, we have the clone JCVI-syn3 with the whole 8 genes, the whole set of genes. And then here we have C3 only with the 79 to up to 181 gene, and this is syn3A. So after 48 hours, both of them were not attached to HeLa cells anymore. We couldn't find any one of them attached, differently from the syn3B with the gene cluster as well as for syn1. So here what we find is that those genes were not exactly capable of bringing back the capacity to syn3B to contaminate mammalian cells or eukaryotic cells. But although these genes actually brought back the capacity to attach to these cells, so there are some other missing genes in the minimal cell that might be responsible for metabolites uptake or other essential functions that are necessary for parasitation. But so what we can say is that although these genes were not capable to bring back the pathogenic capacity for syn3B, it was actually, but we can say that they are actually essential for the attachment of the mycoplasma's mycoid cells to the eukaryotic cells. But then we decided to perform an immunological assay as well. And then we co-cultivated the cells, the syn1, syn3, and our mutants with the DHL60 and DHL60 cells. So these are our negative controls. They were not differentiated to neutrophil-like cells. So what we saw actually is that all the incubation times that we analyzed, none of them was actually capable to induce phagocytosis in these neutrophil cells, differently from JCVI-syn1 and also from the 178 to 186 clones. Also, the 179 and 101 as well. They had a kind of a certain induction. But what is the most interesting is that the clone with the syn3B with the genes that we added back, the full set actually gave us the highest response. They were capable to have a highest induction into the phagocytosis. So this called out another question to us. Would these cells may be capable to be used as vaccines against Mycoplasma mycoides? So as I said, Mycoplasma mycoides, they are pathogenic cells. They causes disease that brought a lot of significant economic losses in livestock as well, mainly for cattle. In this case, Mycoplasma mycoides, it has a high specificity for goats. So what we decided to do now there's a second step in this work, that all the other data was already published in last year, but now these are the new data that we are putting together to try to understand if we will be capable to use those cells as possible vaccines against Mycoplasma. So what we are doing now in partnership with the University of Bahia in Brazil is actually to evaluate the immunological response of the cells in the presence of the PBMC cells, which are peripheral blood mononuclear cells. So they are mostly composed by lymphocytes and monocytes, which are responsible for the innate and adaptive immune response. So we decided to evaluate those cells. So here, what we did was actually to co-cultivate the mutants of the cells syn1, syn3, and not the mutants together with those cells, the PBMC cells. And here we're just checking out for the proliferation of those. And after 18 hours, all of the cells actually interfered with the proliferation of the lymphocytes and monocyte cell, the mononuclear cells. But this is something that we could expect, even if there is no pathogenicity involved here, because you can see here the wild type, the Mycoplasma mycoides, actually got a higher capacity to inhibit the proliferation of the mononuclear cells. But after 18 hours, all of them. But there is a competition, there are other cells there. So, okay, this might be something that could explain this characteristic, but then we decided to evaluate now, sorry about that. So to evaluate now how this could affect the production of the nitric oxide and also the presence of cytokines. So here we evaluate then the presence of nitric oxide, was measured using the Grease test, and also ELISA to analyze the cytokines production from the mononuclear cells. And here we could see that in a certain way, all of the cells were capable to induce the production of nitric oxide. So showing that they were also capable to induce some inflammatory response. And then for the interleukin-1 beta, we can see some of the responses here. So between 6 and 12 hours, we mostly see some for syn1 and also for the wild type. But then 12 hours, it's funny because the mutants actually, it went down. And then after 18, 24 hours, all of them actually had induction of the interleukin-1 beta, but actually syn3 didn't. It was pretty low, which is what we was kind of expecting. The same way for interferon alpha, the TNF alpha, we can see more or less the same responses, except that 24 hours after it was pretty low. But anyway, we are thinking that maybe 12 and 18 hours could be better time frames to evaluate this, because after 24 hours, maybe the cells actually were normally dying. So, but here, we can see also that the wild type has the highest response, and in 18 hours, all of them actually induce the presence of the TNF alpha. The same way here, okay, sorry, this is just to explain a little bit because of the differences of the time frames, but then here we have another one, which is the EL4. This is the other two, they actually are responsible for the production for the pre-inflammatory. So it induces the inflammatory reaction. But the EL4, actually, it's the opposite. It actually holds and controls a little bit, slows down the inflammatory response from the cells. So here, actually, none of them, in a certain way, were capable to induce EL4. Only after 24 hours that we have some of these, but even though syn3A also, it's very low. But although this analysis doesn't bring us too many responses, because sometimes syn3 is actually activating a response, and sometimes it seems, if you stay way longer, if you check on a longer period, you see that the cells actually are activating an inflammatory response. But when we see the whole picture, of this is a heat map results from all the cytokines analyzed, from all the time frames. So you can see here that actually, and this is mostly for the profile after 12 and 18 hours. So here we have our mutants here, which are those guys here, this and this, together with the wild type and the syn1 clustered together. And it's interesting because syn1 is together with the clone with 179 and 80. They have kind of similar profiles of the inflammatory response. And the clone that has the 108 up to the 186 gene, it was kind of has a similar response from the wild-type Mycoplasma mycoides. But what is the most interesting about it is that the JCVI-syn3 actually has the lowest inflammatory response in comparison to all of them. So this is what we were expecting. So this is agrees what we have found in the phagocytosis analysis. So, but then how it's the response of those cells for the proliferation of CD14. So CD14 is a surface receptor mainly found on monocytes and macrophages. So it functions as a pattern of recognition receptor, especially for bacterial lipopolysaccharides. So, and it's also related to the innate immune response. So we check it out how those cells actually induce the presence of these receptors after 12 and 18 hours of incubation with the PBMC cells. And we can see only the wild type, and syn1 actually was capable to stimulate the macrophage response first and the other strains. But after 18 hours, we see that JCVI-syn3A as well had actually was capable of inducing this response. So it had a later stage, but it was also responsive to the presence of this JCVI-syn3, the macrophages. And afterwards, we also check it out for the CD21. It's also another receptor. It is expressed mainly in B lymphocytes and plays a role in activating the B cells by binding to complement fragments. And it's important for the adaptive immune system. So in this way here, again, after 12 hours, mostly the wild type Mycoplasma mycoides actually was capable to induce this response, but after 18 hours as well, we have only the syn1 got a little bit higher in comparison with the wild type Mycoplasma mycoides. Although all of them here, when we say, oh, sorry about this, when we say syn3B here, is only the mutant that has the whole set of genes. So since it actually gave us better results, we decided to move forward with this analysis on with this clone. So I'm sorry, I forgot to mention this. But in a way, so also the minimal cell didn't respond, neither did the mutant at the same way as the wild type, or neither syn1. But when we look for all the lymphoproliferation, or the increase in the proliferation of the all mononuclear cells present in the culture, all of them were capable actually to induce this proliferation, showing that all of them in a certain way can induce an immunological response. This is actually something that is interesting because this might show us that all of these cells might be functional as a vaccine, although neither syn1 or the wild type would be interesting because we believe that they will bring the animal, how can I say, gonna get the disease, they're ill. So this is not actually an option, but maybe the minimal cell and the mutant that with the whole gene set inserted back might be actually very interesting targets for the development of vaccines. So to make sure about this, what we are going to do in the next step, so this will perform pretty soon. Our animals are already in adaptation, so we might start it, I believe, in a couple of weeks. So we are actually perform an infection assay in vitro, in vivo. So we will infect goats with the whole strains and evaluate the bacterial load, cytokine expression, perform histopathology, immunohistochemistry, and transcriptome analysis from the lung tissues. And after we perform these, we believe that we will be capable to have a better clue in how these cells could be or not used as vaccines. But so far, I have spoken with you using the minimal cell for an application. But now, maybe we believe that the minimal cell also can be used for a construction or even to build synthetic cells from the whole, completely from non-living parts. As I said, our synthetic cell is not considered as a completely synthetic cell. It's just a cell with a synthetic genome. So now we are looking for to get new ways or new methodologies that will enable us to produce a completely synthetic cell. So as I said, the membrane is one of the most important parts for the synthetic cell to be built, and even for the cell for all different functionalities in the cell. So why not to take advantage and exploiting the perfect membrane that JCVI-syn3A has and use this as a chassis to actually receive a completely new genome. So what we are exploring now is the use of the endonuclease ICU. This strategy was already used for E. coli, Pseudomonas putida, Houstonia eutropha. It was being published in this paper at PNAS, showing that actually the endonuclease is functional in order to degrade the completely genome from those bacteria. So this could work as well for the minimal cell. We used this only under the control of the tetracycline, was our first experiment, and actually didn't work well because the ICU endonuclease did mediate the breaks and actually end up being toxic. And the tetracycline promoter wasn't very, how can I say, very efficient to control it. We had a lot of leakage. So at the end, we couldn't actually evaluate the functionality. The cells end up not growing or even knocking out the endonuclease gene. So we decided to move to a different strategy where we use also an integrase. So the integrase now is under the control of the tetracycline, and the endonuclease gene is upside down. Let me say it's in the different position between the integrase recognition region. So once I have tetracycline, I have the expression of the integrase, and then the integrase will enable the activation of the endonuclease expression. So this is what we have doing now. So we are trying to do this, and we are evaluating this the mechanism of this genetic circuit to be capable to fully degrade my genome in order to be capable to use this as a chassis to receive a new genome. And I hope to acquire a synthetic cell completely made from non-living parts. So first we evaluated the functionality of these cells using quantitative PCR. And here with these results, so here we are comparing our clones without and with tetracycline, and we see that we have to wait 5 cycles to have the similar signal from the non-induction cell. So this here shows us a 97% of efficiency of the endonuclease, how can I say, cutting the target gene, the specific region of the genome where it cuts. So this gave us very good results. Okay, so the endonuclease is functional, is actually cutting the specific spots in the genome of the minimal cell, but it's actually how can I say, actually inducing the fully complete degradation of the genome. So then what we did was this, let me see, I'm not sure if this will be capable to be shown. I don't know if I'll be capable to show to you. Can you see this, the video?
Yes, the video loaded. It's not playing yet.
So here what you see is our minimal cell induced with tetracycline and of course with the genetic circuit of the endonuclease and stained with DAPI. So this is a time lapsing. So showing that we are actually having genome degradation at the end. So this is very, very interesting movie. And what we performed afterwards. So are you back to the presentation?
Yes, we're back to slides.
Okay. So let me see if I can, again, presentation mode doesn't work perfectly. So I'll go back to the slide viewer. So here what we see is actually a decay of the fluorescence of DAPI showing the degradation of the genome. And let me see, let me go there. Oh, now it decided to work. Can you see this?
Yes, you're in full
Okay.
screen now.
No, I am. Can you only look at this? I don't know why, but that's okay. All right. So this is the move. I don't think it will. Okay. No, I don't want to play. That's fine. How can I come back now? I won't be able to come back now. So okay, so let's move forward. And okay. So, but here what we did actually was to do a color change unit assay where we see actually the growth of those cells under the tetracycline induction. So here we have our syn3B as a control with and without tetracycline. Again, one of the clones, another clone, and here just the integrase inducing the expression of mCherry, just to show that the integrase is not interfering on the metabolism of the cell and neither to the growth of the cell. So here we can see the growth of those cells after 24 hours, and actually we don't see growth at all in those cells. But after 48 hours, we still see, unfortunately, we see some growth. So thinking about that, we have an efficiency of 97% of the endonuclease cutting the cleavage site. Well, these 3% of cells actually are surviving somehow, and unfortunately growing and overcoming my zombie or simple cells population. So now we decided that we needed to actually to better understand and try to come up with a strategy to purify our zombie cells or our simple cells that also call those kind of cells. So we decided to try to do cell sorter using flow cytometer analysis. So what we did actually was stain our cells with DAPI and also FM for the membrane. And what we thought was, okay, we have double positives here where we have DAPI, we have the DNA, we have the membrane stain, but the guys that only have the membrane stained, well, those are my zombie cells, and these are the population that I want to sort. So this is what we did, actually started to evaluate those cells and trying to identify the best gate to perform the sorter. And then here we can see after 6 hours of induction, we can see that the only FM positive cells were increasing towards my clones that were induced, but the syn3B almost didn't have many changes. But the double positive as well wasn't very different. And the same way, when you see 12 hours again, well, the population increased a little bit, and after 24 hours, we went up to 40% of the cells actually only with FM positive. So if you see the kinetics here, just checking the percentage of the cells, here we see syn3B, only with FME positive and DAPI. So we have a larger, they continue growing. And here we have a decay from our cells induced after 24 hours. But the opposite happened with the only FM positive cells, which increased up to 40%. But actually, although this gave us some answers regarding the full genomic degradation, it didn't make us very happy because, well, it seems that only 40% of our population of our cells are actually turning to zombie cells. But this also questions a lot of things. Are we having all the cells actually being marked correctly? Should we try other markers for those cells? Maybe DAPI can be still staining some of the cells that are actually being degraded, but we didn't wait too long or something's not working. But we still have some questions to be answered. So what we are also trying to do is focusing on another strategy to purify our zombie cells, is using the Kasugamycin antibiotic as a selection tool. It blocks the beginning of the translation of the mRNA. So if you have any survival cells, this will interfere with the translation of proteins and everything. So these cells wouldn't be capable to actually double themselves and they will end up dying. And it seems to be working actually when we see 24 and 48 hours after. When we treat with Kasugamycin, we don't see any growth at all. Which is what we were expecting. But at the same time, we are not sure if this is also affecting the zombie cells, since it affects the translation process. So, but it depends on other, I'm going to say, other metabolic pathways. Like once the genome is degraded, I do believe that this won't interfere much to the zombie cells, mostly with the live cells. But actually now what we need to see, it's actually if this is also, if it is really interfering with only the live cells. So what we can conclude so far, well, okay, our genetic circuit actually is functional and time dependent, reaching 97% of cleavage efficiency after 24 hours post-induction. And we have so far, at least what we were capable to see in our flow cytometry analysis, that 40% of our population have the full genome degradation process happening. So what we are actually doing in this moment? Well, we needed to better understand the ISO impact at JCVI-syn cells metabolism. So we are also performing transcriptomics and metabolome to see and which genes are being induced, what is happening inside the cell, and actually how the cell is surviving. I think this is one of the most questions that I would like to ask. So the minimal cell, which just tiny little genome in comparison with the wild type mycoplasma, is actually being capable to escape of this. So what's going on? Which are the genes that are responsible for this? And of course, we need to improve the purification methods of the mycoplasma syn cells or zombie cells population. Something that I also would like to perform is to evaluate for how long those cells without genome are actually functional. Until when? 1 day, 2 days, 3 days. For E. coli, it can stay functional up to months under frozen in -80 and 1 week in room temperature. So we need to understand this better as well. And of course, we are also going to perform transplantation into the cells, the ones treated with casugamycin, the non-treated casugamycin, and of course the regular cells without the genetic circuit for genome degradation, to see if you can, which are the differences between those strategies, if it's working or not. So we are pretty close to have these answers. So with this, I thank you for the attention. I would like to acknowledge everybody that is working with me and collaborate somehow, and the funding institutions here in Brazil, for JCVI mostly, for all the help that and discussions that we are having regarding this work. And I also would like to acknowledge the Synthetic Biology Association, the Brazilian Association of Synthetic Biology, who actually designed these figures here, which I really, really like, and shows a little bit of the SynBio made in Brazil. So thank you very much again, and I'd be willing to answer your questions. Thank you.
Thank you. Thank you so much. That was really awesome. There are questions in chat. Can you see chat?
Oh, yes.
Can you read a question out loud, because people watching the recording don't see the chat?
Okay, so I think the first one is José Fernando. Could minimal mycoplasma be used for mimicking other pathogens, engineering it to present antigens specific to another bacteria? Yes, it is possible. There are some other works that have been done before. There is a group from Japan, Miyaka, I think it's Miyaka, his name. I'm not sure. Maybe I better not say. I might be mistaken, but actually what they did was put back some genes related to spiroplasma, which are bacteria that are capable to have some movements. And actually they brought back those movements to the minimal cell, which is very interesting. So yes, we do believe that this is possible. And it was actually, we have some proof of that from other works that have been done. So the second one is, does a minimal cell have any kill switch or auto-auxotrophy? No, it doesn't. But this is something that we are also thinking as a possible strategy in order to destroy then the survival ones from the zombie cells or the stem cells that we are developing. This could be something that might work. When you do genome degradation in our zombie cells, what's the half-life of mRNAs? So how long after genome degradation those genes stop expressing? So this is what you need to answer as well. Something that we might use, maybe it's a plasmid that will not integrate into the genome, so it won't be degraded, maybe expressing M-cherry. We were using also a metabolic pathway that produces ATP, but I believe that just the fluorescent gene might also help us to answer this question. But the transcriptomic data, the mRNA sequencing that we are performing now, we are going to do it in different time frames. So I also believe that this will give us some answers as well, but I can tell you right now. And last question, could you make self-free extract from the minimal cell, would then express the minimal genome? Yes, there is a John's and Kate is also performing this. So yes, there is some cell extract that have been produced from the minimal cell. And this is another strategy that's coming up as well in order to build synthetic cells from non-living parts.
Thank you.
I think that's all here in the chat.
I think that's all the questions in the chat. Yes. Thanks so much again. I really enjoyed it. I, work with some of those minimal cells myself too, and I can really appreciate the technical difficulty of everything you're doing.
Yes. Sometimes you say, no, this might not be possible, but then but then you have a clue here and there. Yes, it's very interesting. The minimal cell is actually a very interesting chassis for different purposes, either for trying to understand genes that we are not aware about their functionalities, or maybe understand metabolic expressions and how it works and how it interferes in the cellular metabolism, and as well to try to build other stuff as well. So yes, for sure.
Right. Thank you so much. Thanks so much for an excellent talk. And thank you for answering all the questions. And have a
Thank
great
you,
rest of your
Kate. It was a
Monday.
pleasure.
It was a pleasure to have you. And bye, everyone.